With the diffusion of automated serial section electron microscopy techniques, the rate-limiting step in the pipeline leading to the generation of biological three-dimensional models has moved to image processing.
Our protocol provides a step-by-step guideline to produce a dense reconstruction of image volumes in just a few days, combined with an approach for quantitative measurements using three-dimensional models.
With this technique, 3D structure of tissues other than brain can potentially be analyzed to identify structural impairments typical of certain diseases and to improve the diagnostic strategy. Segmentation is a tedious step. Take the time to make it as accurate as possible to avoid proofreading a bad segmentation and having to restart the whole process.
The design of the software is sometimes not very user-friendly. Therefore, it is important to see the procedure to efficiently start work on the segmentation.
Open the image stack by dragging and dropping the native file from the microscope containing the stack, or by dragging and dropping the folder containing the whole image stack into the software window. Once the stack is opened, go to Image, Properties to make sure the voxel size has been read from the metadata. Transform the image into 8-bit by clicking on Image, Type, and select 8-bit. If the original stack is acquired as different tiles, apply stitching within Track EM2. Create a new Track EM2 project using New, Track EM2 blank. Using Track EM2 embedded functions, segment structures of interest if needed. In the Track EM2's GUI.